Thanks for joining us today. Hello, I'm Fenella Saunders. I'm the Director of Science Communications and Publications at Sigma Xi and Editor-in-Chief of the Society's magazine, American Scientist. It's my pleasure to introduce the first of a series of special distinguished lectureships that Sigma Xi is hosting in response to COVID-19. I'd like to acknowledge the Committee on Lectureships, chaired by Dr. Mark Imhoff, for selecting a group of speakers who will share their expertise and engage in conversations on a different aspect of the COVID-19 pandemic. Before we move on to today's session, I'd like to just let you know that the next session is scheduled for Friday, September 11th at 2 p.m. Eastern Time, and will feature Dr. Michael Osterholm, professor at the University of Minnesota and director of the Center for Infectious Disease Research and Policy, speaking on the COVID pandemic, the evolving reality. Recordings will be posted on the Sigma Xi website at sigmaxi.org/lectureships, and keep an eye out there for upcoming talks as well, because this is an entire series. Today, please use the Q&A box to submit your questions. Dr. Peeples will speak for about half an hour, and then we'll do some questions after that. A selection of your questions will be answered by Dr. Peeples after the talk as time permits. So today, I'd like to introduce today's speaker, Dr. Mark Peeples from The Ohio State University. He is principal investigator and member of the Center for Vaccines and Immunity and the Research Institute at Nationwide Children's Hospital, and a professor of pediatrics and of molecular and cellular biochemistry at The Ohio State University College of Medicine. He is also a Sigma Xi past president and fellow. His talk today will be on what vaccine options are most likely to protect us against COVID-19. Thank you for joining us today, Dr. Peeples, and please go ahead.
Thank you, Fenella. I appreciate it. I've been a Sigma Xi member for a long time, ever since graduate school, which is over 40 years ago, and I've enjoyed the many programs that you have. I've been studying viruses ever since graduate school, since the first day of graduate school. Actually now I've kind of, as far as COVID goes, I've graduated again, but this time it's into the high-risk group, which is a little over 65. So I wanted to kind of take you through some things that we've learned and think about when we're talking about COVID-19 and SARS-CoV-2, the virus that causes it. But I wanted to start with a little history. And this kind of takes me back to my 6th birthday. You see me there in bed with a cake, 6 candles, and you can see that my arm has a bit of a rash, and my chest and my cheeks. That was measles. So this was my measles birthday. At the time that I got measles, everybody got measles, all the kids. The only good thing was you could miss school for a couple of weeks. But the bad thing was you felt crappy. You can see here that my parents were a bit of jokesters here. They gave me the game of cootie for my 6th birthday. What I didn't know in this picture when it was taken was that about 4 years earlier, measles virus had been isolated. And about 5 years after this picture was taken, we had a vaccine. And very quickly, measles epidemic every year that we would have declined to almost nothing. So it's just an amazing story for a virus that at one time was a major childhood problem. And it wasn't just that kids got sick and felt bad for 2 weeks. Some kids died. There was an encephalitis that's caused by measles in some kids, and also lung disease. So getting rid of that was really important. It was very important to have a vaccine. But that was all done in 9 years. When I went to grad school, I started working with a different virus, which was respiratory syncytial virus. And that virus is a virus of very young children, particularly between birth and 2 years up to 5 years is where it's the worst. So it turns out that it was in the same group as measles virus. So I was studying a virus in that region. The difference was that here it was a respiratory virus, whereas measles is more disseminated. So I collaborate with some terrific people here at Children's in Ohio State. We're working on a vaccine for RSV. So what I didn't tell you was 3 years before my picture there, RSV was also discovered, and it's been over 60 years now, and we don't have a vaccine. So it's a much more difficult problem. In some ways, it's more similar to COVID-19 because it's trying to protect the respiratory tract. And that's difficult to do. It's more difficult than other organs. So our work here is to try to make a live attenuated virus vaccine that's safe and immunogenic for kids. We're not the only ones working on a vaccine for RSV now. It's become a hot topic in the last 10 years. But I have some terrific collaborators here, my clinical collaborators, Octavio and Asun and Jen Ron here, as well as the others. But the four of us are all working on several COVID projects also. So we're not only working to try to make vaccines that are going to be better than the ones that are coming out in this first round, but also working on the clinical disease. What does it look like in children? What happens when children are infected both with SARS-CoV-2 and with RSV, or in later years, flu, which is more prevalent in teenagers. So there are a lot of things going on here that we're working that are relevant. I've studied RSV for so many years, but when SARS-CoV-2 came along, you couldn't just ignore it. And so we've switched over a lot of our efforts. There's a similarity here because both viruses are RNA viruses. Both of them have envelopes, and both of them infect the respiratory tract, at least to begin with. RSV remains limited in the respiratory tract. It's possible that SARS-CoV-2 goes further into the body. So this is where everything began in China, and now 24 million people are infected, 820,000 deaths, societal disruptions, economic disruptions, and we clearly need a vaccine. So what would be the goals for a vaccine? Well, first, of course, we want to protect against COVID-19, the disease, and we want to do it for at least a year. That's the minimal requirement. It's similar to influenza, where we give a vaccine every year to protect. There, it's partly because the virus changes every year. Here, we don't know how much change there's going to be. But we definitely want a vaccine that will last at least for a year, but it would be better if we could get many years, a long-lived vaccine. So, and a third thing, I would say, because this is a respiratory virus, we want a vaccine that gives us an immunity that protects us from the disease, because we may not be able to protect ourselves completely against the virus. That's certainly what we know for RSV. Once you've had RSV, you've recovered, you can get infected again, but the next time it's not as bad. You have circulating antibodies or T cells, depending on how close the two infections are together. So the good thing is there, when you get the infection, you get a boost. So your immunity that you got from your vaccine could be boosted by a low-level infection by the virus again. So it's not all a bad thing. In the end, we're aiming for herd immunity. We want a certain percentage of people to be immune. We don't know what that percentage is. For measles, it's somewhere 90 to 95%. And I've seen estimates as low as 50% for SARS-CoV-2. I doubt that. I think it'll be at least 80. So that means you need 80% of the people to be protected before you can protect your whole community. So that's critical, particularly for kids who can't be vaccinated because they have an immune deficiency. So we want to be able to protect them too. And the ultimate goal, of course, is no more COVID disease. And we hope to eventually be able to reach there. So I'll start off just looking for a minute at our immune responses. What kind of defenses do we have to protect us against this virus or any virus? First, we have interferons. So when a virus infects a cell, those cells produce interferons, which go to the neighboring cells with an alarm message, such that when the virus comes out of the first cell, it doesn't find such fertile ground in the others. So that can help slow the infection, slow the spread, and slow the disease. People who don't have interferon genes have a horrible time with any kind of virus. The other thing interferons do is they stimulate the B and the T cells. So that'll get things going for our adaptive immunity. So the antibodies are what are produced by the B cells. And when they're produced against every viral protein. Most of those antibodies don't do us any good. The only ones that are helpful are the ones that bind to the attachment protein or the fusion protein. Those are the ones that will neutralize the virus. And even some of the antibodies that bind to that protein don't neutralize. We'll look at that protein in a minute. The other arm of the immune system are cytotoxic T cells. So these take care of virus-infected cells. So they use a very different mechanism and actually go in, find those cells, and kill them. So we have antibodies to take care of the free-floating virus that comes out of the cells, and we have cytotoxic T cells to kill the cells that have the virus, to try to stop that infection. So here, first we have to get to know the virus. So here's this beautiful picture from the CDC where you see the gray envelope around the virus, that's a membrane. And you see one major protein sticking out, and that's the spike protein. And it's a trimer. If you look carefully at the picture here, you can actually see here 1, 2, 3 components. They're all sort of in a circle. So it's a trimer, but each of the monomers in that trimer are identical. We'll see a little higher definition in the next picture. But this is the one that sticks out on the outside of the virus. And when you look at it from the side like this, it kind of looks like a corona. You see these big proteins sticking out from the membrane all the way around. So that's why it's called a coronavirus. There are a couple other minor proteins in here, the M protein and the E protein, but we don't think those have anything to do with neutralization. The only thing that has to do with neutralization is this attachment protein. And why is that? Well, it has two functions. The first is to attach to the cell, and the second is to cause the membrane here of the virus to fuse with the membrane of the cell. So this is like two bubbles. One is a huge bubble and one's a tiny one. That's the virus. The cell is the big one. And when they come in contact, which would be occurring because of the attachment protein, binding to its receptor, which is ACE2 on the cell, and then causing those two membranes to fuse such that the membrane of the small bubble becomes part of the membrane of the big bubble. And in doing that, it spills all of its contents into the cell. That's exactly where the genome wants to be and replicates. And as it replicates, then takes over the cell, makes many more copies of this virus, exports them to infect many other cells. So it's a very rapid spread. But this S protein is interesting because it has both the attachment and the fusion function in the same protein. That's true for some viruses, but not all. So here we're looking at the same protein, but this is now in a molecular model. We actually know what the structure of this protein is from some very impressive cryo-EM studies that were done very early in the epidemic, before it was even a pandemic. But what you can see here again is the trimer. Here you have a light blue, that's one of the monomers. and here's a purple or violet here. They're kind of wrapped around each other. And then the yellow one is the third trimer, but at the far end of that has this red domain. And I've highlighted in red because this is actually the receptor binding domain. So this is the most important part of it. Antibodies close to this part of the protein can, or on it, can actually block infection very, very effectively. So these were the three sort of tabs that you saw in the last picture, sitting right at the top of the virus, of the S protein. If we look at the side here, you can see the same kind of thing. You see the purple here in the back, violet. You can see a little bit of it here. This light blue is curling around here, and the yellow all the way around here. And then part of that yellow is this red that goes up here. So the red is actually like a little trap door, and it will move up and move down. And it's only when it's in the up position that it can engage its receptor ACE2. So it's, in a way, it's protecting its receptor by hiding it and then just letting it out to sample to see if there's an ACE2 around where it can bind or not. It's a pretty amazing little protein. Once this protein does engage the receptor ACE2, the next thing that happens is much of this top part of the protein falls away and exposes the bottom part. And that's where the fusion peptide is and all the machinery that will be able to reach out and grab the neighboring cell and pull the membranes together and cause them to fuse. So it does that by refolding this protein dramatically. It becomes almost twice as long. It's anchored down here at the bottom, and part that's not in this picture because you can't work with the protein very well that way, is a highly hydrophobic domain that goes through the membrane. And at the end of that is a little cytoplasmic tail that hooks up with the internal parts of the virus when the virus is assembling. So this whole protein is standing as a spike on the virus surface. And this form of the protein is called the prefusion form of the protein. That's getting ready to work. So once it binds to a cell receptor, it springs into action, refolds, unfolds, refolds, and brings the membranes together pretty dramatically. Okay, so what do we want to do for a vaccine? The main target for vaccinology here is the S protein, the spike protein. And that's because of this central role. And in fact, antibodies to the S protein are the only ones that neutralize. So what can we do? Well, we could inject it. So we can inject it like influenza virus, the HA protein, and that's what protects us every year from influenza. Like we just talked about the prefusion and the postfusion form, but really the antibodies to the prefusion are so much better, probably 10 times better. That's the way they are in RSV at least. They're 10 times better at neutralizing. So you want to have antibodies to that prefusion form. So like I said, the trimer is anchored in the membrane, and there are ways to make sort of a mini virus. It's called a virus-like particle. So it has an envelope, which is lipids. It buds from a cell surface, just like the virus does. And it has the glycoprotein, the spike protein in the outside. So this is one way to make a vaccine is a VLP. So there the S is still anchored in the membrane. But like I said, the S protein has to make these conformational changes. And for that reason, it must be unstable. We call it metastable. So it's got to be sort of in this in-between state that can spring into action when it's needed. So because of that, one thing that can be done is to stabilize that protein. And in fact, in order to get those crystal, or not crystal, but cryo-EM structures I just showed you, what had to be done was to stabilize that protein. So the way the group did it was to change some amino acids that were at an angle where the protein made a bend. And if that bend was about the right angle for proline, which has a bend in it naturally, they would replace the amino acid at that bend with proline. And that keeps the structure, but doesn't allow the protein to change conformation. So that could be a better antigen. It certainly is better for structural studies like we just saw. The third thing that you can do is to actually replace that anchor with a self-trimerizing peptide. And that, in addition to adding the prolines, then can form a stable structure that could be used as a vaccine. Now, all the things I described here, as far as making that stabilized protein, were really first laid out clearly with RSV, the fusion protein of RSV. And in fact, Jason McClellan, the scientist who made the structure I just showed you, also did the same thing for RSV. So again, it's like your past experience can be used for the next epidemic that comes along. So it's been exciting, and actually helps progress much more quickly. The other thing that can be done, and there are some vaccines in trials now like this, and that is to grow the virus and then to kill it with formalin, and then to use that virus to inject into a person. So that would contain the S protein. It would also contain some other proteins of the virus, and it's possible it could have some host cell proteins also. So when we think about killed viruses, inactivated viruses, so there is actually a very negative story from RSV. The first vaccine that was tried for RSV in the mid-60s, that was what was done. The virus was grown up, killed with formalin, and put into kids' arms in a trial. When they did that, it was fine. The kids had no problem. But when RSV infected them a couple months later, then they had major problems. And instead of 5% of them going to the hospital, 85% went, and 2 kids died. So it was a terrible experience, and set back RSV vaccine work quite a while. We don't really know exactly why that happened, but there are a number of theories. In any case, when you do use an inactivated virus approach like this, you want to be very careful to test it and make sure it doesn't have any untoward effects. So how else could we get the S protein vaccine going? How can we present it rather than just making a protein or a virus-like particle? We can actually inject something that will produce the protein. So one of the more popular ways to do that right now is to use messenger RNA. And the good thing there is that messenger RNA can be made synthetically. It's clean. It has no other adventitious agents. So that may work. DNA also is used sometimes for vaccine. But probably the vaccine right now that's furthest along is an mRNA vaccine. Second kind of approach is a live attenuated virus. So that would be taking SARS-CoV-2 and either passaging it in culture many times, like was done with measles, mumps, rubella, polio, to make an attenuated virus that can be put into a person, doesn't cause disease, but induces a great immune response to all the viral proteins. That's the real advantage with using the whole virus. Of course, we don't have time now in a pandemic to do that. But luckily, we know a lot about these viruses and about enzymes of similar viruses where we can make modifications. And those modifications then may not only attenuate the virus, but have it induce a greater immune response to whatever it's carrying. So I think in the future, we'll see some of those things tweaked. Right now, there are a couple vaccines that are progressing rapidly through clinical trials that are an attenuated, well, no attenuated viruses are, but they are vectored. That's where we're going to go next. So the third way to do this is actually to take a virus that's innocuous, like adenovirus, causes common colds, but we have a form of it that's missing one of its genes, such that if it ever gets into one of our cells, that virus can only live in one cell, it can't replicate, and it can't produce any progeny. So it's not going to cause an infection from that virus. So you can build in a gene that you want to deliver, in this case the S protein gene, into an adenovirus and deliver that in a vaccination. So that's probably the second in line in the vaccines that are being developed now. There are other viruses that make sense and that people are working on. One is the measles virus vaccine. That's already attenuated. We give it to kids all the time. And if we built the S gene into it, perhaps we could have a good vaccine. The good thing with measles vaccine is that we know that when you're vaccinated, you're protected for many, many years, decades, if not whole life. So that's a real advantage. That's what we would like. That's a kind of ideal vaccine. So if you could express the S gene from that measles virus, maybe the S gene, the response to it, would also be long-lived. And that's what we would hope. So that's a possibility. Another is VSV, vesicular stomatitis virus. So this is a bovine virus. Because it's bovine, it doesn't infect people real well, but it'll infect a cell and it'll produce a lot of protein. It just doesn't spread very well in the person. So the good thing is it grows to very high titers in culture, so we can make the vaccine easily. And this first came to prominence during the Ebola pandemic, actually epidemic in Western Africa about 3 years ago, where toward the end, this vaccine had been made and was given to people, and it stopped the epidemic. So it really has a good history there and is doesn't cause major problems. So those are sort of our tools, things that we can use and people are working on. Certainly there are probably over 200 groups working to make vaccines at different stages. Now, once you do have a vaccine and you want to do something with it, you want to get it out into trials, you have to manufacture it. So first you have to go to CBER at the FDA and talk to them about your process of production, your process of purification, and what kind of data you have already, what kind of data you need before you can submit an IND for an investigational new drug. And then you've got to scale that up, do all those things in a larger volume to be able to meet FDA standards. So like I said before, the mRNA is chemically synthesized. It shouldn't be very difficult to clean it up. You're not getting rid of biological material like you have to do with the other, all the other approaches that I mentioned. If you're using a protein, though, you do have to produce the protein and purify it, and certainly it would be better if you were to stabilize it so it would remain in that pre-fusion form, so you could get antibodies to that form, which are going to be much better at neutralizing. And then the attenuated virus, you would be, first you have to make sure that it's attenuated, won't cause disease when you put it into a person, and not attenuate the immune response. So you still need a good immune response. And you want no reversion. You can't have a virus like this going into somebody and on some percentage of those people it reverts and keeps the infection going. So those things would all be tested and carefully monitored. And then you have to grow it in cell culture and have some purification. The vectored vaccines also have to be grown in culture and purified before they can go into people. So once you have a small batch for your first trial, you got to test it. So first thing you want to do is in animals, you induce a good, robust immune response. And generally we look for antibodies, number one, from the B cells, but also at T cells to see if you get a response there. T cell responses are different for everybody depending on their HLA. So it's a little more difficult to know how much of a T cell response you'll get in every person, but you can test those things. Eventually, though, you have to prove that your vaccine will protect an animal from challenge with COVID-2. So now this has to be done in a BSL-3 lab. All work with COVID-2 has to be under higher containment. Most of our labs are BSL-2, but BSL-3 is what's critical here. So it's a lot more restrictions, a lot more care in everything that's done to make sure that nothing gets contaminated, nothing gets taken out of the laboratory that's still infectious. So that makes this a bigger challenge. The other challenge with animals is that they don't express, most animals don't express the receptor for COVID-2. For mice, for instance, but a transgenic mouse will work. So there are a couple strains of those that were made actually during the first SARS epidemic that have been frozen as embryos and brought out and are being bred now for these studies. So once you show that you can protect from challenge in an animal model, the next thing is clinical trials. So phase one are often people with antibodies. And what you're looking for here is there any cytotoxicity, any toxicity to the person, any kind of symptoms caused by the virus itself. The second phase is you look for people that don't have antibodies and you immunize them. And usually you do about 20 and then half as many placebos. This would be a small phase 2 trial. And what you're looking for there is, does the virus or the vaccine induce neutralizing antibodies, which you can detect in culture, and then do they protect from infection? Now, how do you determine do they protect from infection? Usually what we would do is let that person just go about their normal business and see after the course of a year, have they been infected again? Have they been protected? Look at people that got a placebo versus the vaccine. Here, we probably don't want to wait that long. And so there may be challenge studies set up to do that last phase of the testing. And then phase 3, you take many more people, 2,000 to 3,000 people, plus half as many placebos. And there you're looking for any kind of rare adverse event. So those rare adverse events aren't seen when you just do 20 people or 200 people. You need many, many more. And so these are the important steps there. And of course, like I mentioned, you could speed some of these up if instead of waiting for natural infection to come, actually take the person and give them the virus, see if they're protected. And there are sites around the country set up to do those kinds of studies under controlled situation where you could be monitoring the patient as well as taking the samples over time to see if any virus grows in the nasal samples, that kind of thing. So that will certainly be done. Now for COVID-19, because it's such a push and such a rush, some of these things have been combined. There are some phase 2 trials, larger phase 2 trials that are being combined with the phase 3 to get things going quickly. If there's no evidence early on of problems or toxicity, they're racing ahead. The other way to speed this up, of course, is if you have a lot of money, is to do the manufacturing along the same time as you're doing the testing. And of course, that's risky because what if you get to the end of the testing and you find there are some objectionable side effects, either with the original vaccine or when they became infected after that. And if there were, then you'd have to throw away all those vaccine doses. On the other hand, the calculus has been made, at least here in the US and some other places, that it's worth the challenge, that we need to do that. So that's what we're doing. Here now, coming back to the goals, we want to protect against COVID, and you certainly want to do it for a year. All the vaccines that are going to progress to certainly phase 3 trials should be inducing a good, strong antibody response. You may want to stabilize the protein to get even a better antibody response. And the vector will be producing the soluble or the membrane anchored protein, one or the other. But really, I think the challenge is going to be protecting for a longer term. I'm pretty sure that one of these vaccines that are being worked on now, or at least one of them, will be useful for short-term protection. But how about long-term? We will induce memory no matter what the vaccine is. But the antibodies that go up can come down very rapidly too. So like with RSV, they go up and within 3 or 4 months, they're very low again. That doesn't mean you're completely unprotected. You still have B cells, and if you get reinfected, they're called up much more quickly, and they respond and can take care of that exposure. But yeah, so live attenuated vaccines really have, I think, the advantage for longevity because you can get all the proteins of the virus are there and can induce a T cell response also. So some of the last cells, the long-lasting plasma cells have been found in bone marrow. So those are actually producing antibody for a long, long time. And certainly we know measles can protect lifelong. That should be our goal. Just the wild cards, how can we make better vaccines, those kinds of things. We need vectors that themselves induce a better immune response. We're working on things like that. The purified or the vectored expressed S proteins can be manipulated to make them more immunogenic by stabilizing them, like I mentioned, by aggregating them so that they're more reactive with B cells, by removing glycans, perhaps exposing neutralizing epitopes, adding glycans to hide distracting epitopes, and adding adjuvants, which can greatly enhance the immune response. And again, herd immunity is what we're looking for, 80% or more. The last thing I would say is we don't want to be cutting corners. An incompletely tested vaccine could be a terrible blow to vaccinology overall. In fact, if it's not completely tested, many people won't take the vaccine to begin with. And an incompletely tested vaccine, if it does cause health problems, either during immunization or upon natural infection, that could lead to people refusing to take any COVID-2 vaccines. And there are already many people who are against taking vaccines, and they may forego even this, and that problem may expand. We certainly don't want that. So here's a picture of my lab. About half of us work on COVID and half of us on RSV and the other half on both. A couple of people here are gone, but that's most of us. So I'd be happy to take any questions if anybody came up with any during the talk. Thank you.
Thank you. Thank you so much, Dr. Peeples. It's been a really interesting talk. We've gotten a number of questions. We will not have time probably to address all of them. So I will just initially say to participants, if your question is not addressed, I suggest you maybe post it again on one of Sigma Xi's social media channels. We are live tweeting this talk, so you can post it there, and we'll try to see if Dr. Peeples can answer additional questions later. One question that came up that you might like to address, Dr. Peeples, is what is the significance of the ACE2 receptor binding? Initially, it was thought that the ACE inhibitors affected the progression of the disease. Has that been proven?
Well, as far as I know, the ACE2 receptor, the ACE2 protein is a transmembrane protein on the outside of certain cells, and those are the cells that can be infected, the target cells. And it's actually an enzyme that is a protease. That's its normal function. As far as I know there are some studies to try to block that receptor and perhaps protect people. I think the problem with that is if you do that, you would also block the protease activity. And it's a protease that cleaves hormones to make them active. So that could affect other physiologic systems. So I think that's kind of a tricky thing to do. It's certainly something you'd want to test in animals before you tried anything in humans.
Another question we've received is, will a mixture of mRNAs coding for the various viral surface proteins work as well as an attenuated virus?
I don't think so. I think the problem with RNA is that, well, first of all, it doesn't replicate. You don't get any, you get one message and it can make a bunch of proteins. That's true. But if you have an adenovirus, it can produce lots of RNA and continue to do that for actually quite a while. Or if you have the actual attenuated virus, it'll make its own RNA. There's probably a limit to how much RNA you can put into someone. There are some mild reactions during the immunization. And so if you wanted to get others in there, you'd have to be upping the dose of RNA. And I think that would be a problem. So far as I know, it's only been the spike protein that's been in the mRNA that's been used.
Another question we've received asks if you could elaborate a little bit more on the mechanism behind mRNA vaccines in terms of are cells taking up the rogue mRNA, or how does that work?
Yeah. So it's kind of like a technology that we use in the lab all the time, transfection of either plasmids or RNA. So essentially you sneak the RNA into the cell, and once it's in the cell, it goes to the ribosomes and is translated. And the more RNA you get in, the more protein you get made. And these, as far as I know, are intermuscular, so it's into the shoulder. And so this is really a fairly new idea. It's been around for, I think the first time I heard of it was maybe 3 years ago. And there are a couple of companies that have been developed based on it. the good thing about it is you can take any virus that you want to protect against. Once you have the sequence, you can make that mRNA and you have a vaccine. So it's a very rapid response time. My concern is that the way it's being expressed and the low level of protein that's made may allow just, a response and protection for a short period of time. I doubt that there'll be a long term. But where we are right now, I think we would welcome a vaccine that we could take even every year to keep us safe until we have a better vaccine that lasts longer.
So there's another question here that actually taps on something that came up in the news this morning about people who are attempting to make nasal delivery vaccines. And there is some point in that news story that those might be more effective than intramuscular vaccines because they offer potentially immunity at the point of infection. So this question is asking about if those delivery mechanisms will be more effective. One thing I noticed in the news report is it didn't discuss the fact that a few years ago an influenza nasal spray was proven to be ineffective. So that might also be something to consider in your response if there's been nasal spray vaccines before and what effectiveness they've had and if that applies in this case.
Yeah, well, intranasal delivery is certainly a way that this could go. The vaccine that would be used there most likely, well, first you have to have some sort of vaccine that gets into the cells in the airways. And here the best vaccine would be a coronavirus vaccine itself, an attenuated coronavirus. I have not seen yet clinical trials on an attenuated SARS-2 vaccine. There may be some out there, but there are other hurdles to overcome to get to that point. For RSV, for instance, that's the only way we even consider delivering that vaccine, to get to the airways, because then you get B cells made in those areas, and you can get secretion for up to 6 months of antibody that can help protect that airway. Still, after about 6 months, that dies down, and the antibodies in the serum disappear with a half-life of a little less than a month. So they're gone. So for a season, for instance, if this were seasonal, the problem is COVID-19 doesn't look to be seasonal. For RSV, we know it goes season is from October, November to March or April every year, just like flu. So we can vaccinate people against flu right before that, like right now. We expect we'll be able to vaccinate people for RSV at the same time, intranasally. And that will induce that immune response and have it still there, active in the area when the infection comes along. But the other approaches, the adenovirus and others, I believe, are all given intramuscularly. So part of it is you want to get the best response is number one. And number two, you want it to be in the right place. Even when, if you have a whole body immune response because you've been vaccinated in the arm, you can still be protected. There'll be a response right after your infection that'll bring immune cells into the area to take care of the virus. So this was a big question with polio back in the early days, because the first vaccine was intramuscular. It was the killed vaccine, but it ended up working very well compared to the oral vaccine, where the idea was, well, you make IgA in the gut where the virus replicates, and that should be even better. It didn't really turn out to be that much different in real life. Yep.
So we may only have time for one more question, possibly two, depending on the length of your answers, but something that multiple people are asking is if there is more then one vaccine that comes out and is available, will people have to choose, or is there any problem with taking more than one vaccine simultaneously?
Oh, I hadn't thought about that. No, I don't think there's probably not a problem, except the problem is going to be there's not going to be enough vaccine for everyone. So I think the idea would be you get one vaccine, and that's probably it, at least per year. That's what I would imagine. But actually, that's interesting you say that. So when I was a kid, polio vaccine came out when I was a bit older, about 11. And so my mother marched us up to the doctor. We each got 3 shots, 1 a month, and we were protected. Then the live oral polio vaccine came out. She couldn't find it in our town, but the town we used to live in was giving sugar cubes every Sunday, first Sunday of the month for 3 months. We drove an hour and a half each way to get all of us vaccinated again. And the reason was my father had had polio when he was a kid, and he was still suffering from problems in his legs. And my mother wanted to make absolutely sure that never happened to us. You look at that reaction compared to some of the reactions you see today to vaccines, it's sort of night and day, where people are refusing vaccines that could really be helpful. We need this to be more of a public health imperative. So I don't think there's going to be an overabundance of vaccines right to begin with anyway.
Okay, we're getting very close to the end of our time. We might be able to squeeze in just one more if you can answer it quite quickly, Dr. Peeples, if you wouldn't mind. And something that a couple of people have asked. They're actually wondering why the measles vaccine is so effective compared to maybe some other types of vaccines, and concerns about vaccines in this case.
That's a great question. I think what it is measles infects dendritic cells, and dendritic cells are the ones that set up the whole immune response. So I think right from the start, it's being produced in just the right place, the lymph nodes or the spleen, where the T cells and B cells are being educated. And I think somehow that is involved with getting such a good, strong immune response. It's not all the answer, but I think that may be part of it, the localization. Great question.
We've actually had a number of really, really good questions that we are not going to get time to. So I thank everybody for putting them into the chat. If your question was not answered, again, I encourage you to please send them to us via social media, Twitter or Facebook or some other way. If you are a Sigma Xi member, you can always put them in the communities as well on the Sigma Xi website. Dr. Peeples has been answering as many as he can, but we're just not going to be able to answer all of them. So please do get us your questions via a different channel before we run out of time here. Thank you again, Dr. Peeples. This has been a really fascinating talk. And I just want to close by thanking again the Committee for Lectureships, and to remind you that the next talk in this series of distinguished lectureships about COVID-19 will be on September 11th. You can find more information about that on the Sigma Xi website under the Lectureships tab, and that this video will be recorded and posted there if you have missed any part of it. So thank you so much for joining us. Thank you again, Dr. Peeples. And we are out of time today, but please do come back and join us again next time.
Thank you.